Okay, here we go. Is that so? How does it look there? It's probably small. Okay. How is it now? Readable? A bit more? Okay. Yes and no. It's going to be a lot of Well, I can do it a bit more. Right. So thanks everyone for choosing this workshop. Ooh, that's nice. It's mostly going to be a package demo. If you have an account on the Galaxy workshop, Galaxy, what's the short? Workshop.bioconductor.org. Then you can sign in like this. And once you're signed in, I have uploaded the content of this workshop in the EuroBioC 24. You'll see package demo iSEE here. Clicking on it, you can click on Run Tool. And when you run tool, you'll see a message. Started, I did a job to the queue. There you go. And then when you have the link open, you can click on that link and it will open an RStudio instance with the material preloaded. I'll try and follow here, see how it behaves. And I've got my local session if things crash. But yeah, so the workshop materials are in this RMD document, which is already open. And rather than going through the text version of it, I have pre-compiled it into a notebook, which is a little bit prettier to look at. I will be expecting that you're familiar with the Summarize Experiment class. For the sake of time, I don't have the sound. Oh, okay. Let me.
It says talking, but I can't hear anything.
Oh, how do I? No, this is not the meeting, right? Where's the meeting? Oh, here. Your screen sharing audio. It's speaking of my voice. I'll try there.
Okay.
Oh. No, well, I switched to another microphone and back to this one. Is it working? Okay. Yeah, so Summarize Experiment and Shiny as well, which is the CRAN package for interactive web applications. This workshop is the successor of at least 4 other workshops that are listed here. So if I don't cover some of those are much longer than today's. So you may find some slightly outdated information in those, but all the features that used to exist still exist, plus more. So I'm encouraging you to ask questions at any time. Don't wait for the end so that the question is in the flow. If you have questions and we're applicable, we're a bit short on time, but I can try to live code something to demonstrate a feature that I didn't anticipate. And if you want to ask questions also or not interrupting the flow, feel free to type it as an issue during or after this workshop. These are the packages that are involved. So iSEE is the core one, and I'll be talking a little bit about the others in due course. The first 10 minutes, I'm already eating into them, is to show you the core functionality, and then I'll go into some advanced things like what extension packages are and how you could write one yourself. But yeah, so I want to show you that iSEE allows you to interactively explore omics data. I want to show you extension packages that exist, and I want to understand. I want you to understand what's needed to write an extension package. We wrote one in about 2.5 hours on Monday, Federico and I, but that's because we know how it works in the first place. Be patient with yourself if you try the first time. So Summarize Experiment, again, I said I expect you guys to be familiar with it, but we all know that Summarize Experiment contains assays with features in the rows, samples in the column. The samples can be single cell. If you have reduced dimension coordinates, you will most likely use a single cell experiment object with the extra slots. There's metadata on the rows that are stored in, and on the columns, which are stored as separate data frames. And that makes the Summarize Experiment a beautiful data structure to work with because it's nice, it's predictable. A lot of things can be automated when you can predict where the data is stored. And that's what iSEE does. It basically knows where to sniff around for assays, row counts, normalized counts, and so on, and just basically builds you a Shiny app where all the drop-down menus are pre-populated with all the possible choices you can dream of. I want a UMAP colored by gene expression and shaped by this and shaped by that. It's all, you don't have to worry about writing the Shiny app. Now, yeah, usually you would be building your own summarized experiment from your raw data. Here, we're going to save ourselves a bit of time and just load a data set that's slightly pre-processed. Where are we? Here we go. Is it going to pop up here? Well, since they're loading the package.
You want to put it on the right side.
Put what?
The console on the right side so that you have most product.
Oh, I'm very old school in using the thing.
Go to the knit button.
Square, knit button. Oh, this thing.
Yeah, no, yeah.
And console on right?
Yeah.
All right. The font bigger? Yeah. Is this enough or do I need more? Well, it's just so it's still readable. It's all right? Okay. So we've loaded a data set. It's 20,000 genes in 379 cells. We've got one assay, which are the row counts, and we've got, so the row names are the gene symbols, the call data are some QC statistics, and so on. And the cells have their just boring database identifier name. Currently, we don't support alt experiments. I don't know if there's any alt user in the room, but this is on the roadmap once we get a bit of funding and therefore time to work on it. This is all, 90% of iSEE has been developed grant-free. And I'll get back to the little bit of money we got to develop some of the extension package. So yeah, basically running iSEE is you have a single experiment object, you load iSEE, you load Shiny just to get the run app function, and all you do is iSEE of my object. What does iSEE do? It grabs your object, it scans everything that's in there, and it's populating this dashboard that's going to appear anytime now with a bunch of panels that we've given very sort of elegant names, very structured names. So the raw data table allows you to see the raw metadata. There's basically nothing there. The feature assay plot allows you to look at a feature expression, so that the assay value for feature. Column data plot looks at the sample metadata, so the column-wise. Sample assay plot, you have one sample and you can plot something about it. In this case, the counts. So every dot is a gene, and you have the counts for that gene in that particular sample, and so on. Column data table, that's the sample metadata, which you get all the shiny goodies about filtering the table. And the complex heat map initially shows only one gene to minimize the loading time. And we could talk about adding more genes to the heat map, which is going to be nicer later. So all those panels, you don't need all of them. You may be interested in only one of them, and we'll get to how can you customize the app to specifically do what you want. I think that's the idea of going through. I don't need the launch meeting. That's what I want. Yeah, so the core functionality, sorry, I've gone back to this. So view the tables of metadata, filter the rows, selecting rows, and so on, viewing plots of assay data, of metadata. And yeah, I guess the heat map where it gets interesting. So I've got this, the pop-up from Zoom. So you have a heat map, you're like, how do I put my genes there? So we have for every panel a data parameter thing where you can actually say which features do you want to showcase in there. There you go. So you can type them by hand, but that's going to get boring pretty fast. What's more interesting is selection parameter, receive my row selection from the sample assay plots. Now you can see that something popped up over here. The other panel says, oh, I need to transmit my selection to the complex heat map. So you go there, you make a selection, and that guy selects, and and now you need to disable the custom rows over here. And suddenly the heat map says, hey, I'm receiving selection from that panel, and I'm showing you the expression. Now, again, we can talk all day about how do you make this heat map look better? Do you change the color scheme? Do you scale? And so on. So we've got loads of options all over the place, which in this case likely to be in transform. Genes being on different scales, you usually want to center so that they're at least back on the same scale, but even that's not enough most of the time. You want to scale them so that they're all between -5 and 15, which, yeah, so this is centered just because you got so many negative small values that actually the scale is uneven. But yeah, the detail of how you actually configure or how you play with the app is very specific to your data set, to your research question. But the point here is that basically every panel you have here can be customized, can be played with and told to display something else. So this is a functionality. So you can draw selection, you can transmit selection, you can use those transmitted selection to highlight those points in the receiving panel, or you can actually restrict the plot to say, show me only those points so that you can actually focus on your favorite cluster, zoom into it, and ask more specific questions in downstream panels. You can rearrange the layout of the panel. So we've got a bunch of, so we've tucked away a bunch of generic options up here. So organized panels will tell you, oh, I want my raw data table to be after the other panel, and I want it to be wider. So you apply the settings, and then the thing gets reorganized. You can stretch the panels to use it. It's, you know, it's not the fastest app in the world because it's the R layer on top of the JavaScript and everything, but we've made it as flexible as we possibly could and got the Shiny contest most technically impressive prize for it. So the Shiny people were impressed. So I think we're pretty happy with ourselves. So you can resize in width and in height. You can download the panel outputs. This is also a fun one. So if you see those plots, you say, well, I want it as a figure in my paper, or whatever. Download the panel outputs. You have the list of all the panels. You select the one you want. You download. It's going to download it as a zip file, and then you have those things to play with downstream. You can launch interactive tours. I'm not sure it's going to work out of the box here. Yeah, so this thing, you can configure it to basically teach your users, oh, this panel does this input UI does that. The default tool is only enabled for a specific data set so that it's predictable. So in this case, it's not working out of the box, but
we want to display that every panel has a mini tour.
Oh, and
show what they can do.
Yeah, this little question mark here is actually working because it's very sort of generic about each panel, what each panel can do. The data parameter box, and it lets you click on it. So it's a guided tool where you're expected to take actions to really follow it entirely. So, and all this can be configured by users. So as a user, you can just worry about someone serves you the app on a web page and you play with the app. But as a developer, you can actually take our package, make your own app, and you are the one showing your data to other users. Yeah.
Like disclaimer or text as
Yeah, well, so you'll see that in the extensions, anyone can add any kind of panel to the interface. If the most generic thing is saying, oh, my panel can't receive or transmit any selection, it's just like a read-only panel. So you can pretty much, I think, configure anything. And if you can't, you just let us know, and it's about us opening an API, one more function. But we've done a markdown board, basically, where you can type up your notes as markdown and click a button, and it renders it on, no, not even click the button, it live renders it into a nice, pretty HTML. Fede?
What would you need a disclaimer for?
I mean, like, this is, for example, if I share iSEE with the users, just to remind them, hey, this is this data set, or genes are case sensitive, something like as simple as that, but just put it on as on the page, so when they want to watch it, they see it as a reminder.
Yeah, if you wanted to open at lunchtime, so the tour, if you define a tour, the tour automatically opens when people open the app, and the first message of the tour could be your disclaimer as well. So we've used that. We've got a gallery. So I'm getting out of the app now, but I've made a website where we have a gallery of apps, and you can see, like, I think the first example here is basically their tour is not about how to use the app, it's about a welcome. This is what the app is about.
If you want, you can just crank some content in there. If you don't need it, you just click away, and then let's say the ones that never saw that, read it.
You can export. So that was our favorite one, and probably one of those that impressed our studio folks. Extract the R code, generate a script that if you copy and paste that script in your R session, that script regenerates every output of every panel. So you get not only the output that you've played it interactively, but you can also grab the code, reproduce it exactly, or take that code and say, tweak the thing that you didn't like that we did. So saying, oh yeah, I don't like your color scheme. I'm gonna grab we're giving you the code and you go away and fiddle with it. So that's the selection, the shiny brush. And here that feature assay plot took the expression of that particular gene in the top hat counts, stuffed it into a data frame, added a bit of thing, and then that's all the code that the app has run in the background.
So it's not a guess from ChatGPT.
Yeah, it's not reverse engineer. It's actually forward thinking. We save the code as we execute it. I realize I'm actually going into way more detail than I expected here, but launching custom interactive tour, opening vignette, inspecting the session info, learning more about iSEE, how to cite it, and Federico actually had a object metadata. It's not in the core app, so
because it's not in
yeah,
yet.
Exactly.
It will be there.
About this data set, and if you click on this, then you have, so the metadata slot, not the column or the row metadata, but the object metadata, you actually unpack it as JSON format. So if someone stuffed the name and the address of the lab and the list of co-authors, you can find that too. Okay, so I'll probably close this. Yeah, I need to close the app. Yes?
And the feature sample, I think it was really useful.
Mm.
So was it possible to do a statistical comparison?
So I think, yeah, you'd probably have to implement a custom panel.
Yes, but we kind of opted for analysis first, exploration/visualization only afterwards. Like, we want to put stars. We don't want to put stars on the plots because otherwise, let's say it would count as a test in the implicit way of the hypothesis that we've been testing. It's an exploratory thing, so like it wouldn't even count. Whether you want to have a feature assay plot that also on top of the, let's say on the MIA plot also does some testing, you could implement it.
Yeah.
We've never felt the need of that.
Yeah, Antonino, correct me if I'm wrong. Doesn't Bigomics, the Explorer, do some tests in the app? It does. So this is one of our sponsors for the conference. And yeah, I've talked with them in the past. So that was one of the discussions I had with Ivo. Is that, oh yeah, does iSEE do that? I said, we initially chose to not do any calculation during the app. It's just data framing things that are already calculated for pure visualization.
Not many, but some.
Yeah. And eventually, once we got the core thing in place, we opened the door for plugins that you can make the panel calculate whatever you want, but if it takes 10 minutes to calculate, I'm not recording it interactive dashboard anymore. Because the app takes a second or a few seconds to refresh, but if it takes 10 minutes to calculate, it will take 10 minutes to refresh.
Yes, I understand that.
Yeah.
So for me, I think if you have a new data set and in your cancer type, you just want to see how does that gene expression work on your subset of interest. It would be just really good to see, hey, look at BRCA1 where you know your genes are just, oh, great, fantastic. Let's do the statistics and you move on.
Yeah.
I'm not saying that you can eyeball the p-value, but say if your eyes are trained well, you'll see whether there is a hint of a significance or so. Take the code, add one line with Gigi's plot or something, whatever that does, and then probably you get all the things and even more than what you need.
Yeah.
But,
love it.
Okay.
Thank you.
So now what I've done during that time, because it takes like a few seconds to run, is calculate. We only have the count, so I've log normalized them. I've run a PCA, a t-SNE, I've added a bit of metadata. And if you do that, notice that the next bit of code is exactly the same as before, iSEE of the object. But the object has updated and iSEE will detect that. So if you run it again, suddenly we have one more panel that we didn't have before, which is the reduced dimension plot. We didn't have any reduced dimension EPC and stuff. Now that there is one, iSEE is like, hold on, there's one. Why don't I show you the panel that displays it? And there you have it. So it automatically detects what we teach it to detect, which is very easy for summarized experiment. And yeah, that's the PCA. And if I go back in the data parameter, there's PCA, and I can switch to t-SNE, and it switches to t-SNE.
Can you color by phenotype?
I can color by. Do you want to color by phenotype? Do you so this is not a phenotype. This is a QC metric, but phenotype would be the core type, or no, maybe not the core type. The driver 1S. Yeah, so here is like 3 conditions, and so you can color by whatever you want. And the code tracker will keep updating and tell you the code to generate this plot or that plot. You can shape by whatever you want. You can size by something, or actually just increasing the size. The point size, I want it to be 3. So either it's something, static, or it's something that's related to your data. You can fast set, which was also a fun one to implement. So you can fast set, oh my God. Okay, we have to look at that one. Why am I getting it? That's not the moment for a phone call. But yeah, so, okay, interesting one. We run into those once in a while. I will have to rerun it to show you what I meant, or I can just show you directly. Yeah, so iSEE, when you launch it like that, it enters what I call showcase mode. It's putting one panel of every type that's compatible with your data. If there's a reduced dimension, it's going to put it. If there's not, there is not. However, it can, if you have a big data set, loading 8 panels every time you open the app is a bit costly. So we have an argument called initial, and you can say only show me an iSEE app. Show me an iSEE app that starts with the initial panel that's just a reduced dimension plot. And maybe just to make it a bit interesting, I'll throw in a feature assay plot, which seem to be popular today. And so when you launch that, I have an app that loads faster because I only have 2 panels to show. And those panels, if I say what you were saying before, like color, make it Tisney and color by driver 1S. Oopsie. There you have it. I go to my extract the, is it extract? No, it's not extract the R code. It's display panel settings. I haven't covered that one yet. That one is another piece of R code that defines the initial I've just manually defined. So if you copy that bit and, so where's my, oh yeah, I need to close that bit. I run this. So it defined the word initial, which is now the list of panels, the list of settings for my 2 panels. And I can say as iSEE SCE initial equal initial. And it's going to do the same job. So my app is going to reopen in the state that I left it in last time. So that's how you can serve an app to people saying, I'm not showing you a black and white app and telling you how to click around your way to get to the interesting stuff. I'm showing you the interesting stuff the moment you open my application. Right, so theoretically 5 minutes left. So I'll just, so this is the idea. So you play with the app, and then you grab the script that defines the initial with all the parameters. Some of them are relevant, some of them you've changed, some of them are irrelevant because we're storing the entire state of the panel. So if you haven't sent a selection in, there's no point defining some of the other parameters. So you can do it by hand after a bit of training when you get used to the app. Initially, borrowing the code that's reported by the app is the quickest way to get you started. And yeah, so that was the screenshot. So if you launch it in initial, you get your app. And that's also, if you want to, that's exactly what I just said. Right. So that is like core functionality of iSEE that was supposed to take 10 minutes and I've taken the whole time. Existing iSEE extension. So iSEE works on its own, but it only defines 8 panel, the ones I've listed before, all the way up to the heat map. Then we started way early in the project, we already got requests, oh, I would like to calculate differential expression, or I would like to do this, or I'm working with pathway analysis. Can I throw in a pathway in the heat map rather than all those things? So that's where we started playing. So ICU was actually the first one, the ambitious iSEE universe, where we basically use a package to prototype a bunch of panels, including the markdown that I mentioned before. Our first volcano plot was there, MA plot, and so on. So it was a little bit of miscellaneous things prototyped together. And then we entered a little bit more, a more structured way of scoping those functionality within packages. So ICD is for differential expression. So that's where you'll find the new volcano plot and the new MA plot, what we call the log FC, if you want to compare the log FC in 2 contrasts, and so on. IC hex, well, it's still very prototypish. It's one of the early one where for large data set, I wanted to summarize them in hexagonal bin using the xbin package. It works, but it opened a whole can of worm about how do you want to summarize discrete data, continuous data, and so on. So let's say it's not final, but the idea is there. iSEE Hub, for those who know the Bioconductor Experiment Hub, is a portal to the Experiment Hub listing all the data sets in there. And then when you click one, you can actually say open in iSEE, and it will download it from the Experiment Hub, open it in iSEE. Caveat with that, there's initial configurations, they pretty much all come in default configuration, which can be a bit slow sometimes. And iSEE Hub was the prototype of iSEE Index. So what I really wanted was a portal where anyone can plug any data sets anywhere on the internet. So the Experiment Hub was taking part of the problem away from me, saying, yeah, Bioconductor is hosting the data set. I have an R function where I can fetch any of them at the touch of a button. iSEE Index was more like, okay, this person is storing it on Zenodo, that person is storing it here, that person is storing it there, and everyone should be able to basically fetch the data, build a summarized experiment object if it's not in that format yet, pass it to iSEE, and iSEE does the rest. So at the moment, we're supporting like basically HTTPS, as long as it's public, and AWS, even private ones. So you just need to configure all your password and accounts, but you can fetch a private data set behind a password connection. And yeah. Or local. If you have a file already on your computer, you don't need to upload it anywhere just for the app to re-download it. You can actually point to the file path on your own computer and it's going to load it. So the way iSEE Index looks is like this. So you have and that's also someone asked for customization, but I'll talk about this later. So available data sets. So this is like a shiny table. You click on the one, you click on configure and launch. Here it's going to open and reveal a new tab where you can even, the maintainer of the app can even offer you different startups, initial states, saying, oh, I want to focus on figure 1 of that paper, or I want to focus on figure 2 of that paper. And it will launch iSEE app potentially on the same or on a different data set in different configurations. So yeah, and I basically allowed people to add a header and a footer to the website where they can put whatever they want. And this is actually me taking Markdown to the extreme and saying, actually, you don't need to just describe it. You can throw in an image in there as well. So because it's shiny and it's completely open source and you can mess with pretty much anything you want.
But you can think of that is basically when you're done with the publication, then you're publishing a couple of data sets, so you can have just one landing page for all the data that you can contain. And
yeah,
I would say that editors and readers of your paper might love it.
This is one paper, and each app focus on one figure, but you can basically have anything you want. And so this iSEE Index was actually where I was lucky enough that a company in the US approached me and to work for them as a consultant for a year. They wanted that package and basically hired me to do it. So yeah, academic funding is not the only way. So that's there. Input data. So this is what, if you wanted to see iSEE pathway, I'm probably going to skip running the code and just scroll through it now. Airway is a nice little experiment with 2 conditions where you can run some differential expression tests in it. So 63,000 genes in 8 samples. A little bit of cleanup because this is the treated level. The reference level is treated instead of untreated, so I just fixed that. Then I am using the OrgDB to basically turn those ensemble ID into gene symbols and make the things readable to a human. Run dseq on it, then you have your dseq table with your fold change and p-values and so on. And then I had to basically, because iSEE wants a summarized experiment, I had to find a way to basically get those differential expression results inside the object. And so what I'm doing is I'm using the raw data slot and I'm nesting those table of D genes in there. If you've tested a subset of genes, I'm just going to populate the rest with NA so that it fits the dimensions. But the differential experiment data needs to be embedded into the object. You can spot it here into the raw data. So this is not just one column of raw data, it's a nested data frame. Then I go one step further. I take that list of DE gene and I run pathway analysis on it. So this is just like a bit of brute force pathway analysis. I'm grabbing them from the OrgDB package and I'm running FGCA on it. again, this is not a lesson about pathway analysis, so I'll skip over the detail, but for each pathway, you've got a p-value, you've got an enrichment score, and so on. And then what iSEE pathway does, again, you need to embed those pathway results in it. Now, pathways don't fit the rows, which are the features, or the columns and samples. So here I'm hijacking the metadata of the object to stuff them in there. But because all the functions like talk to it, well, they've been developed together, they know where to expect the information. And yeah, once you've done this, I log normalize because I don't want to show you counts. And when you launch the app, I'm launching the app with a pathway table, a volcano plot, and a complex heat map, which again, I would have to run all the code to make this visible. So in the pathway table, you can select a pathway. And when you select that pathway, so all the genes in that pathways are actually transmitted to the other panels. So in the volcano plot, it's hard to see, but you got some dots in red where I've highlighted where the genes of this pathway are in terms of differential expression. And so this one is receiving the selection. Same thing for the complex heat map. It's receiving the genes in that pathway. And then there's this extra panel for those who know GSCA analysis. So this is receiving the pathway from here. And within that pathway, I can see where the genes that are in the leading edge, the one driving the enrichment, are located. And actually, this is the selection, sorry, I talked too fast, that I'm sending to the heat map. Because if I was sending the entire pathway, it would be, well, it's not a huge pathway, but it's potentially 100 genes, including 90 of them that are not expressed. So if you send them to this panel at the bottom, you can see what the important genes in that pathway are and focus your heat map on that pathway, on those genes. Yeah? So this is one or two of the extension, ICD and iSEE pathway, working hand in hand to customize the iSEE app and add panels that the base iSEE package does not include. So yeah, that was crash course in showcasing a couple of extensions. You can imagine that like iSEE index would take me another 10 minutes probably to show you, but it's probably the most exciting one that I've done, I think. And writing iSEE extension, so how did I do that? The panels, we've implemented them in beautiful Bioconductor S4 classes. Why? Because it's reusable, it's beautiful. So we have a class called panel, and it defines everything a panel should do. Then under that class, we have a panel, a subclass called dot plot, and another one that's stable. Because if you're showing a scatter plot, you're not showing, it's two completely different things. So dot plot does certain things that a table can't do, and the other thing around. And so we've got this hierarchical structure, which I think I forgot to add here. We have it in an earlier workshop, and you just make your way down until you end up in a final concrete plot like the heat map or the reduced dimension plot. Yeah, okay, I'll wrap just with this. So making a new panel is literally like setting a class that says, I'm a reduced dimension hexagonal plot. I do everything a reduced dimension plot can do, but I have a bin resolution, and then later on, you're going to have the code that actually shows the data as bins rather than dot plots. So this is the easy way, as I call it. You take a panel that's already working, and you kind of overwrite what it does with what you want the new panel to do. If none of the panel are even close to what you want them to do, so the one we've done with Federico earlier, you want to show the list of cells as text in a panel that where you've made that selection. None of the panel do even remotely that. You may have to start higher up in the hierarchy, saying, I am deriving from panel, and then you just have a bit of extra work defining the core things of what a panel should do. But yeah, so make a class and then just customize it. We've had one recent success story, iSEE3, where someone who's not one of the core iSEE developer actually made a whole fully blown package that allows to visualize tree summarized experiments in iSEE, which again, I'm not a tree summarized experiment regular user, so it would have been extra work on me to develop it, while that person had a vested interest and put in the time to make it work.
Yeah, then it's truly like that.
Yeah, there you go. There's your.
Yeah. So it's a proof that it's possible to. You don't have to be one of us to extend that.
You can become one of us. But yeah, So that's okay. Last minute question. We are now 7 minutes into the break, so there's 23 minutes of coffee left. There's more speakers for those who haven't, for potentially back of the room who didn't get the speaker yet. And I'm happy to answer any question now or upstairs or tomorrow as well. Thank you.
Thank you.